All right. So first thing I'm going to go over, the synapse. I'm going to talk about that a bit. Second thing I'm going to talk about are the neurotransmitters. And then the final thing I'm going to talk about is receptors. I'm not going to try to cover everything that's in the chapter. I never do. I'm just picking the most important concepts that students have the most difficulty understanding. These are really important concepts for understanding psychology, for understanding how therapeutic drugs work, for understanding addiction, which is a huge problem, and for understanding psychology in general. So I'm going to start with the synapse. Monday, I already talked a little bit about that. We did the case study talking about the synapse. And when you think of synapse, there is going to be two broad classes. We'll have a so-called gap junction, and we'll have a chemical synapse. Okay, synapse is basically where two neurons connect. And gap junction is common in invertebrate species. So we see lots of gap junctions in them. In humans, it's a fairly rare synapse. In cardiac muscle, we see lots of gap junctions there. Okay, so the gap junction has the advantage of being really quick. So ions just flow through channels from one cell to another. That's very fast, but the effects are short-lived. Okay, sometimes that's good, sometimes that's bad. It depends what you want to do. If we want to modify behavior for the rest of our lives, a gap junction is not going to be real good. A chemical synapse is going to be much better. Right? So this is the most common synapse in us. This is crucial for psychology. All right, so when we talk about drugs of abuse, when we talk about therapeutic drugs, all these things are going to affect the chemical synapse. With the chemical synapse, I want you to think of two broad classes. This isn't in the book per se, but it helps me keep things straight. The first is the acetylcholine synapse at the neuromuscular junction. The second is everything else. Okay, so when we talk about these synapses, the main difference is that at the neuromuscular junction, we're going to use metabolism to terminate the signal. And hopefully you remember from the reading or from Monday, we're talking about acetylcholinesterase. Everything else is going to use reuptake to terminate the signal. Okay, so keep these in mind. If I tell you that I want you to think about the neuromuscular junction, you should not be including any transporters. You shouldn't be talking about reuptake because that doesn't happen at that synapse. If we're talking about a dopamine synapse, there's no metabolism in the synapse per se. You use reuptake to terminate the signal. All right, I'm going to go through the neuromuscular junction first, the NMJ. I'm not going to number these because every time I number them, I've got a different number. It's just kind of arbitrary. Okay, so here is the presynaptic neuron. And then here we have the postsynaptic cell. I'm saying cell because this is muscle. Okay, we have our axon. We got our action potential, comes screaming down here. At some point, we lose the voltage-gated sodium channels. Instead, we have voltage-gated calcium channels. Okay, so remember, gating means they're open or closed. Voltage tells me what opens them. So in this case, it's the membrane potential. And this tells me what flows through it. Okay, so this is just a tunnel through the membrane that lets calcium flow when I reach a certain membrane potential. So when this depolarization hits, and I'm only drawing one, but really there's a whole bunch of them, right? When that happens, calcium's gonna come into the cell. All right, so we haven't talked about it explicitly, but there's a higher concentration of calcium outside the cell than there is inside the cell. So the second these open, calcium comes in. Calcium then begins a cascade of biochemical reactions. And what that is eventually going to result in is one of these synaptic vesicles containing neurotransmitter. In this case, because it's a neuromuscular junction, we know it's acetylcholine. Okay, so the synaptic vesicle containing acetylcholine is going to fuse with the membrane and releases acetylcholine. Okay, so one action potential hits the terminal. Voltage gated calcium channels open. Calcium enters the cell through the channel. Calcium begins cascade of reactions that causes the synaptic vesicles to fuse to the membrane. Acetylcholine is released into the cleft. Okay, once it's in there, it's going to diffuse down its concentration gradient. On the postsynaptic cell, we're going to have a couple specializations. One is going to be a set of proteins called nicotinic acetylcholine receptors. These are monovalent cation channels. So that means sodium and potassium flow through them when they're open. We also have an enzyme called acetylcholinesterase. Okay, so when acetylcholine is released across the cleft, eventually it's going to interact with the nicotinic acetylcholine receptor. So acetylcholine binds to the nicotinic acetylcholine receptor. When it binds to it, it activates it. When I say activate, what I mean is it causes the channel to open. Okay, so these are, and this is a term you'll hear down the road, ligand-gated channels. Okay, so you know what a voltage-gated channel is. Ligand-gated channels means they're opened by the ligand. Okay, so what's the ligand? A substance that binds to the receptor. For these nicotinic acetylcholine receptors, it turns out there's actually many ligands. One of them we're talking about now, one ligand is acetylcholine. Acetylcholine is going to bind to these receptors and activate this. What do you think another ligand for it is? Well, based on the name, right? Nicotinic, you all recognize the word nicotine, I'm sure. I hope. All right. So nicotine also binds to these channels. Okay. But here we're talking normal physiology. Normally, there's no nicotine in your body. Right. So acetylcholine binds to the receptor and activates. Sodium flows in to the postsynaptic cell. That's key to remember where you are. And potassium flows out. You know that sodium wins. And the postsynaptic cell is depolarized. At the neuromuscular junction, depolarization equals contraction. So as long as acetylcholine binds, sodium is going to flow in. I get a depolarization. This means muscle contraction. Okay, I'm running out of space here. So I'm going to move up here. Okay, so depolarization equals contraction. Acetylcholine eventually gets broken down by acetylcholinesterase. Okay, so this acetylcholine molecule is going to move around It'll hit that receptor, it'll open it up, some sodium will come in, I'll get a depolarization. It'll then bounce off and move around a bit more. It'll go to this one, open that one. Eventually, it's going to interact with acetylcholinesterase, though. When it interacts with acetylcholinesterase, acetylcholine goes away. Okay, so without ACH, the nicotinic acetylcholine receptors close. Depolarization goes away. And then finally, muscle relaxes. Okay, so there's a lot here. You should be able to walk through this in your head. That's what you'll need to do on the exams, but more importantly, for your future, that's what you're gonna need to be able to do. So we've got a good start. When we think about this, anything that disrupts signaling at the neuromuscular junction is going to really be a problem. If we have, as we've talked about, if we do anything to interfere with the activation of these nicotinic acetylcholine receptors, the muscle won't be able to contract. And if that inhibition is strong enough, you will no longer be able to breathe. Likewise, we talked about some of the more potent chemical weapons. There's a class of chemicals called organophosphates. These aren't so important from psychology's point of view, but these are poisons, toxins that are in the environment. They're also modified to make biological weapons or chemical weapons. So we talked about Novichok, which is a well-known tool of assassination. Sarin gas, which has been released on the Tokyo subways, for example. These are all organophosphate derivatives that interfere with acetylcholinesterase. Okay, so in that case, without acetylcholinesterase, the acetylcholine stays there too long, the muscle stays contracted, and we get something called tetany. All right, so that's the neuromuscular junction. Now I'm going to move on to what I'm going to call the monoamine synapse. Okay, so in the psychopharmacology chapter, there's a lot of terminology. That terminology is really important. Just memorize it. When it comes to things like monoamines, the monoamines, if you don't remember what that means, you're going to be confused, right? I hope you've read the chapter already, but in case not, here now, I'm going to refresh your memory. But on an exam, I won't. I'll just say monoamine. Okay, so the monoamines are a class of neurotransmitter that are derived from amino acids, right? They're relatively small because there's modification of just one. So this is going to include things like serotonin, which is abbreviated 5-HT, 5-hydroxytryptamine, dopamine, which is abbreviated DA, dopamine, right? And also things like norepinephrine. In terms of psychology, though, we're going to focus mostly on serotonin and dopamine synapses. All right, so the monoamine synapse is largely similar to the neuromuscular junction. So I have a terminal, right? So this is a presynaptic neuron. Here's a postsynaptic neuron. Okay. We've got our vesicles containing monoamine neurotransmitter. We have our action potential coming down as normal. Here we have receptors. Okay, so the structure is very similar. Okay? Just that this second cell is another neuron, not a muscle in this case. So it's not going to contract, obviously. All right, so my action potential comes down. Voltage gated calcium channels open up. Calcium comes into the cell. That starts that cascade of reactions. The vesicle is going to fuse to the membrane and release its content into the synaptic cleft. These are going to then bind to receptors and have a postsynaptic effect. Now, one thing about these receptors, here when we're talking about most dopamine and serotonin receptors, most are metabotropic. Okay, I'm going to go into more detail later about what this means. Okay, but the nicotinic acetylcholine receptor is an ion channel. So when ligand, acetylcholine, is bound to it, it opens up the channel and ions flow through the receptor itself. Here, when these are activated, we get a cascade of biochemical reactions is initiated. Okay, so the receptor itself is not an ion channel. Ions do not flow through this receptor. Rather, when dopamine binds, we get a change in biochemistry. Okay, and this change in biochemistry can do many things. There will be separate ion channels here. Okay, this change in biochemistry may open or close ion channels. It may affect gene expression. Okay, so we can actually turn genes on or off by activating these receptors. Okay, so there's literally hundreds of different effects that my neurotransmitter binding to these receptors could have. And I'll go over the difference between ionotropic, metabotropic, if not later in this lecture, in one I record after. All right, so action potential comes down, calcium, voltage gated calcium channels open, calcium comes in, causes the release of neurotransmitter. That neurotransmitter is going to bind to and activate receptors for it. That's going to initiate changes in biochemistry of the cell that may result in ion channels opening or closing. It may result in changes of gene expression, many different things. All right. Now, again, we don't want this signal hanging around forever. We need to terminate the signal. Okay, so there are special proteins called transporters. Okay, these transporter proteins carry neurotransmitter across the membrane. Okay. Now, usually they're going to grab neurotransmitter and bring it into the cell. Okay. However, there are some circumstances where that transporter will actually carry neurotransmitter from inside out. One of the effects of amphetamine is it reverses the dopamine transporter. So instead of dopamine in the synapse being brought into the cell, dopamine is actually grabbed from inside the cell and kicked out. Okay, but usually we're going to grab the neurotransmitter and bring it into the cell. Once it's in the cell again, now it can't interact with the receptors. Okay, so this process is called reuptake. And this is a really important process. Many drugs of abuse target these transporters and affect it. So things like cocaine, methamphetamine are going to alter function at this synapse. Now, the name of the transporter is dependent on what it carries. Okay, so you need to know a transporter is a protein that carries neurotransmitter across the membrane. So if you hear me say something's a transporter, I expect you to remember what it does. Now, there are specific types of transporters. I'm not going to go through them all. There's one, often abbreviated DAT. This is a dopamine transporter. Okay, so no secret, we're not trying to hide it. What do you think the dopamine transporter brings back into the cell terminal? Obviously, dopamine. Okay, there's also one for serotonin. So we'll often talk about the 5-HTT. 5-HT is serotonin, T is transporter. So serotonin transporter. Now, these are important, again, for many, many, many reasons. A lot of therapeutic drugs antidepressants, things to help with OCD, anxiety disorders. Many therapeutic drugs target this transporter. And what they actually do is block it. The dopamine transporter is more related to drugs of abuse. Okay, so things like cocaine, methamphetamine. By the way, I will spend a little bit of time talking about methamphetamine. Part of the reason for that is Southeast Asia, including Singapore, is starting to have a pretty bad methamphetamine problem. Methamphetamine is a nasty, nasty drug, extremely addictive, so addictive that people will kill, prostitute themselves, steal, all kinds of things to get methamphetamine. That's something you do not want in Singapore for sure. Okay. Any questions on what I've gone over so far, either the dopamine synapse or the monoamine synapse? All right, if you have any questions, obviously you can come back to these. I'm actually going to ask you a couple rhetorical questions, and I'm not going to answer them. These are for you to think about later. After I'm done with this session, we'll be happy to go over. I'm going to stop recording, and I'll go over with the people that are still here the answer to those questions if you still have them. Okay, so first question. What happens to the postsynaptic cell if I block the transporter? Okay. 2, what would happen if I got rid of all the calcium in the body? I'm going to give you just a minute to think about that with this diagram before I move on. I'm going to pause recording for a minute, and then I will be right back. Okay, so those are synapses. That's all I want to cover now. The way I drew it out, what I wrote, you should be able to do that also. Right, classification of neurotransmitters. There's many different schemes. I'm not gonna focus on, let me get the dog real quick, I'm sorry. Sorry about that. He's excited about something. All right, so with the classification, there's lots of different classification schemes. We can do it by function. So we can talk about neurotransmitters, neuromodulators, neurohormones. Okay, all of those neuro are in neurons, but they do different things. When we talk about neurotransmitters, we're going to talk about the transfer of information at a synapse, right? So a synapse is a specialized structure, right, with synaptic vesicles, voltage-gated calcium channels. In the postsynaptic membrane, we're going to have receptors. Okay? I'm going to focus about neurotransmitters. I'm going to give you the big picture first, and then I will go into some details about each that I want you to know. Okay, so with the neurotransmitters, there's lots of different types. I'm only going to go over a couple. Okay, so with these, I'm going to talk about acetylcholine. Okay, so acetylcholine is in its own group. Okay, next, we're going to have monoamines. Okay, the monoamines are named based on their structure, like I said. Mono means one. Amines has to do with an amino acid. So all the monoamine neurotransmitters are chemicals that are different versions of modified amino acids. Okay, so in the monoamines, we'll have the catecholamines, right? And the catecholamines, again, are named because of chemistry. There's a catechol group that gets added. That means nothing to you probably. Just know that they're biochemically related. We'll also talk about the indolamines. Again, so these will be amino acids modified with a catechol group. These are amino acids modified with an indole group. Okay. Now, in terms of the indolamines, we'll have serotonin. Another indolamine, it's not really a neurotransmitter, but you've heard of it, I'm sure. It's involved in sleep, circadian rhythms, and some other things, is melatonin. Okay, but when we talk about indolamines, think serotonin. For the catecholamines, there's three. There's dopamine, there's norepinephrine, and there's epinephrine. Okay. In terms of psychology, dopamine, which is abbreviated DA, and norepinephrine, which is NE, are more important than epinephrine. We're not really going to talk about epinephrine much. This is also known to endocrinologists as adrenaline. Okay, so whether you call it adrenaline or epinephrine depends on whether you are an endocrinologist or a neuroscientist, but it's the exact same chemical, just different names. When we look at it as a hormone, it has a different mechanism of action, different properties than when we're talking about it as a neurotransmitter. In terms of psychopathology, things like that, this has a fairly minor role, so we're not going to talk about it very much. Serotonin, as you know, is abbreviated 5-HT. The reason these are important is because if I tell you something is an indolamine, you can tell me something about its synthesis and its chemical properties. Same with the catecholamines. So there's an enzyme called monoamine oxidase. MAO breaks down monoamines. So which neurotransmitters are going to be affected by MAO? Dopamine, norepinephrine, epinephrine, serotonin. Early antidepressant drugs were MAO inhibitors. Okay, so they inhibited the enzyme that broke those down. So an MAO inhibitor is going to increase dopamine, norepinephrine, epinephrine, and serotonin. Okay, so you need to know these names. Monoamine, catecholamine, indolamine. These are words that are used constantly in psychology and especially in neuroscience. All right. In addition to the monoamines, we're going to spend a lot of time talking about a few amino acid neurotransmitters. And here we are going to talk about glutamate. We'll talk about GABA. A GABA is gamma amino butyric acid. I'm just going to call it GABA. You will not have to know what it stands for on a test. You'll need to know all about GABA because it's an important neurotransmitter, right? But don't worry about the chemical For 5-hydroxytryptamine, that you'll need to know, and you'll see why. All right. So for the amino acids, the two we're going to spend time talking about are glutamate and GABA. There are others, but I'm trying to focus on the most important ones for psychology. So pretty much everything I've written down here, you should have committed to memory, and you should develop a working knowledge of it. All right, glutamate is the major excitatory neurotransmitter in the adult brain. GABA is the major inhibitory. Neurotransmitter in the adult brain. You see, I'm being careful, right? I'm talking about the adult brain. That's because during development, glutamate is actually inhibitory and GABA is excitatory. But in you and me, glutamate will be excitatory, GABA is going to be inhibitory. Okay. Finally, there are a couple neurotransmitters that are gas. The book calls it gasotransmitters. I don't know if I like that name, but we'll go with it. The main one we'll talk about here is nitric oxide. Notice that it's nitric oxide, not nitrous oxide. Nitrous oxide is the stuff they use at the dentist. People call it laughing gas. It's used as a propellant, like in whipped cream cans, things like that. Right? That's nitrous oxide. Nitric oxide is different. It's a completely different chemical. Nitric oxide is important. It's so-called retrograde neurotransmitter. Okay, so the way all of these neurotransmitters work is they are released by the presynaptic cell. They travel across the synaptic cleft and stimulate receptors on the postsynaptic cell. Nitric oxide carries info from the postsynaptic cell to the presynaptic cell. So it goes backward, right? Retrograde. Okay, we will talk about nitric oxide down the road when we get to learning and memory. It turns out this is going to be pretty important for learning and memory. Any questions on what I've talked about so far? This is just memorization, right? Just stuff for you to memorize. Now, I'm focusing on neurotransmitters. Okay, neurotransmitters are released by a presynaptic cell, carry information across the synaptic cleft, interact with receptors on the postsynaptic cell. If something is a neurotransmitter, you know a lot about its mechanism of action. You know about what it's doing, how it's working. There's another major class of neurochemical the book talks about called the neuropeptides. Neuropeptides have a different mechanism of action. They're involved in a lot of important, more physiological functions. We will talk about neuropeptides down the road, but I want to focus for now on neurotransmitters. All right. couple things I want to point out. I'm going to talk about genes. Okay, so the definition we are going to use is it's a stretch of DNA that codes information to build a protein. Okay, we are making new discoveries all the time. This definition of a gene is changing as we learn more. But this is a good basic definition. Stretch of DNA, of course, I mean sequence of bases. Again, depending how much chemistry you know. All right, so genes code for proteins. If it is not a protein, there's no gene for it. Okay, this is key. Remember this. I can't emphasize this enough. Is there a gene for intelligence? Yes or no? Type it in the window. Is there a gene for intelligence? Keep typing, everyone. Type in no, 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 right? According to the lay press, maybe what you've been taught before, you think, oh yeah, there's a gene for intelligence. No. Intelligence is not a protein. There is no gene for intelligence. How about homosexuality? Is there a gene for homosexuality? No, 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 no. Good. How about schizophrenia? Just keep typing no, 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 no. How about addiction? Okay, no, 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 no. Good. This is a really important point. There are no genes for these things. There's no criminal gene. Okay. Now, if we think about intelligence, there are genes that code for proteins that affect neural function in such a way that intelligence is affected. Okay, so this is accurate. There are genes that code for proteins that affect the way your brain works. The way your brain works can be measured in terms of intelligence. Okay, so it's key to remember there's no gene for intelligence, but there are certainly tens of thousands of proteins that affect how your brain works. Okay, good. All right, so this is key. Everyone here seems to be on board about it. When I ask you this on the exam, you guys will all get it. All right. Let's think about dopamine. It is a monoamine. It is not a protein. Is there a gene for dopamine? Good. If I don't have a gene for it, how do I get dopamine? How do I regulate it? The level of dopamine. Okay. So what I'm going to go through next is something that's very important. This is a concept. Okay. So you need to memorize the specific, yes, but understand that this concept applies to many other neurotransmitters. Okay. Neuropeptides, oxytocin, vasopressin, insulin, these are large proteins. There are genes for those. For neurotransmitters, small molecules like dopamine, glutamate, GABA, no gene. All right, so I'm going to show you the synthetic pathway of catecholamines. Again, what are the catecholamines? Dopamine, norepinephrine, and epinephrine. Okay, just need to memorize that. I'll keep saying it, so the more I say it, the more you'll remember it. Okay, so we don't have a gene for dopamine. What we need to do is synthesize it. We need to build it. All catecholamines are going to follow a similar pathway. Okay, and this is actually in one of the figures in your book. What I write down, I want you to remember. Okay, so everything I write down is what you need to know. So the first thing we're going to do is get tyrosine. So tyrosine you can get in your diet. So we get tyrosine primarily from our diet. Tyrosine is going to get converted into another chemical called L-DOPA. Okay. Now, if I take a bunch of tyrosine in a dish and I let it sit there for 10,000 years, I'm not going to get any L-DOPA. This isn't a reaction that just spontaneously happens. Hopefully you remember from basic bio, if we have slow reactions, we can speed them up with enzymes. Okay, so an enzyme is a protein that speeds up reactions. Okay, so normally this wouldn't happen, but we'll have an enzyme. I want you to know the name of this enzyme. It's called tyrosine hydroxylase. I will refer to it as TH. So when you hear me talk about TH, I expect you to know that it's tyrosine hydroxylase and that it is going to convert tyrosine into L-DOPA. Okay, this is a very important enzyme for psychology. All right, L-DOPA you probably have never heard of. If you have a family member or know someone with Parkinson's disease, you may have heard of L-DOPA. L-DOPA is sometimes given as a treatment for patients with Parkinson's. We'll get to why down the road. All right. L-DOPA is going to get converted into dopamine. Okay. Now, I don't want you to remember dopa decarboxylase. Just know that there's another enzyme. Okay. Now, if I want to make norepinephrine, I'm going to take that dopamine and I'm going to convert it into norepinephrine. Okay, there's an enzyme called DBH, dopamine beta hydroxylase. Okay, again, just know that there's an enzyme that's going to take dopamine and convert it into norepinephrine. Norepinephrine will then get converted into epinephrine if we want. Again, there's something called PNMT. You can just call it enzyme. Okay, so these enzymes, you need to know that they exist. You don't need to worry about their names, okay? The reason you don't need to worry about their names, but you need to know tyrosine hydroxylase, is it turns out TH is the rate limiting enzyme in catecholamine synthesis. Okay, and so what do I mean? Well, in order to make dopamine, right, I have to take tyrosine, convert it to L-DOPA, and then that gets converted into dopamine. What if I want to make more dopamine? Well, I need more tyrosine. I need to convert it into L-DOPA faster and convert that into dopamine faster. What really determines how much dopamine I make is how much tyrosine hydroxylase there is. Okay, so unless you're malnourished, and none of us are, unless you're malnourished, you have more tyrosine in your body than you need. Okay, the amount of tyrosine does not limit how much catecholamine you make. Okay, so you've got an excess. Okay, now TH is actually what is regulated. Okay. This enzyme here, dopa decarboxylase, there's tons of it around. Okay. So if there is any L-DOPA around, there's so much of this enzyme, any L-DOPA floating around is going to get converted immediately into dopamine. Okay, so this conversion will happen very quickly and easily because there's so much of that enzyme around. Now, I need to make an important point here. We're talking physiology, okay? Physiology is not like money. Okay, and what I mean by that is if I have a little money, that's good. More money is even better. A ton of money is even better, right? More money is better, 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 better. Okay, so we always have this mentality, more is better. In physiology, that's not true. In physiology, you need the right amount. Okay, what happens if your body temperature, we'll start with Singapore. What happens if your body temperature goes up? It's 40 C. You're out in the sun working really hard. What happens to your body temperature? It goes up. What happens if it keeps going up? You die. Okay, so if body temperature gets too high, you die. Let's come to Buffalo now. Right now it's like -5 C out. All right, so if we go outside and we're not properly protected, into the freezing cold. Our body temperature is going to drop. What happens if your body temperature drops too much? You die. Okay, so if our body temperature is too high, we're dead. If our body temperature is too low, we're dead. Where does our body temperature have to be? The right amount, around 37°C, right? Plus or minus a few degrees, obviously. Okay, so we need the right amount. When we're talking about neurotransmitters, receptors, all this stuff, more is not better. Okay, we need the right amount. Okay, so get that in your mind. Okay, so we need the right amount of dopamine. If we have too much, we're in trouble. If we don't have enough, we're in trouble. Okay, so the way your body deals with that is we control how much TH is around. Okay, remember, we've got tons of tyrosine around. Okay, we've got tons of this enzyme around. So if we do have any L-DOPA, it's going to quickly get converted into dopamine. Okay, so what happens? Let's say I've got normal amount. Okay, so I've got some tyrosine hydroxylase around. It's going to grab whatever tyrosine it can from the environment, and it's going to convert it into L-DOPA. That L-DOPA is immediately going to get converted into dopamine. Okay. What would happen to the amount of dopamine if I decrease how much tyrosine hydroxylase is around? Well, there's still plenty of tyrosine. There's still plenty of this enzyme. That's not changing. With less tyrosine hydroxylase, though, I'm going to be able to make less L-DOPA. Okay, so if I decrease the amount of TH, I'll have less L-DOPA. That L-DOPA is going to get converted right away into dopamine, but there's less of it. Okay, so less TH means less L-DOPA. That means less dopamine. What if I increase the amount of TH? So now I've got a lot more TH. Well, now we can take all that tyrosine and convert it into L-DOPA. Again, there's so much of this enzyme around that the instant we make L-DOPA, it's going to get converted into dopamine. Okay, so it's this TH gene that's tightly regulated. This is going to determine how much dopamine we make. Now, I mentioned this before. It's a simplification. One neuron equals one neurotransmitter. There's exceptions, but for the most part, when I talk about a dopamine neuron, it makes dopamine and that's it. All right. If I'm a norepinephrine neuron, what neurotransmitter do I make? If I'm a norepinephrine neuron, I make norepinephrine. Okay. So if I'm a norepinephrine neuron, do I make tyrosine hydroxylase? Well, in order to get norepinephrine, I need dopamine, right? How do I get dopamine? Well, L-DOPA. How do I get L-DOPA? Yeah, convert it from tyrosine. So a norepinephrine neuron makes tyrosine hydroxylase. Good. Okay. If I'm a dopamine neuron, do I make TH? Yes. Right. So if I'm a dopamine neuron, I'll take tyrosine. I'll convert it into L-DOPA. That'll get converted into dopamine. Now, if I'm a dopamine neuron, I want to stop, right? If I make this enzyme, DBH, I'm going to convert that dopamine into norepinephrine. Okay, so how does a norepinephrine neuron make norepinephrine? How does a dopamine neuron make dopamine without norepinephrine? And it has to do with this enzyme. I'm going to use the name just because I'm used to it. This is an important enzyme, so I use it a lot. You just need to know enzyme, okay? So if I'm a dopamine neuron, I'm going to make TH. I'm going to make dopa decarboxylase. But I am not going to make DBH. I'm not going to make dopamine beta hydroxylase. All right, so if I don't have this enzyme, am I going to take tyrosine and convert it into L-DOPA? Yep. Am I going to take L-DOPA and convert it into dopamine? Yep. Without this enzyme, though, am I going to take dopamine and make norepinephrine? No. Okay, so the difference between a dopamine neuron and a norepinephrine neuron is whether or not they make this enzyme. Okay, any questions on that? That's an important point. So all catecholamine neurons will have tyrosine hydroxylase. Only norepinephrine and epinephrine neurons will have DBH. Dopamine neurons do not have DBH. All right, hopefully that is clear. For serotonin, I'm going to simplify it a lot. You're going to take tryptophan, through 2 steps. We're going to convert that into serotonin. Okay, so this enzyme is called tryptophan hydroxylase. Don't worry about it. And this is 5-HTP decarboxylase. Don't worry about that either. Okay, so tryptophan is going to get converted into an intermediate. That intermediate is going to get converted into serotonin. These are done by enzymes. What would happen if there's a genetic mutation and enzyme 1 didn't work? Is there a gene for serotonin? It's a monoamine. It's not a protein, so no gene for that. How do I get serotonin then? Well, I have to make it. If this enzyme doesn't work, nothing else can happen. Okay, if that happens, we'll have no serotonin. Any questions on that? Okay. I'm going to start talking about dopamine. Dopamine is hugely important for psychology. I mean, if you've heard of one neurotransmitter, it's going to be either dopamine or serotonin, right? Anything relating to addiction, though, reward, dopamine. Okay, so I'm going to talk a lot about dopamine throughout the rest of the semester. When we talk about reward and reinforcement, and therefore addiction, dopamine is central to understanding that. When we talk about things like Parkinson's disease, dopamine is essential. In schizophrenia, schizophrenia involves important dysregulation of the dopamine system. Okay, so dopamine is a really important neurotransmitter. We're going to talk about 3 systems. I'm going to give you their names now, tell you a little bit about their functions. Memorize these. Again, there's a lot to memorize, right? All right, so the first will be the nigrostriatal dopamine system. Okay, so here we have the soma are in the substantia nigra. What's funny about this nucleus is everyone from kind of Western New York calls this a substantia Niagara because of Niagara Falls. It's not Niagara though. There's no way. Niagara, substantia nigra. Okay, literally black substance. It's called that because in fresh human sections, you can actually see it with your naked eye. It's stained black. A byproduct of catecholamine synthesis is a pigment. So neurons that make lots of dopamine also make this pigment and it turns them dark. All right, that's trivia that will never come in handy, but all right. The terminals are going to be in the striatum. All right, so you were supposed to know that the striatum is important for movement and includes the basal ganglia. And this is why this neuroanatomy is important, right? Here in this lecture, I'm going to go over it slowly and I'm going to tell you all this stuff. But if you're talking to other people or on an exam, I'm not going to tell you that. This is stuff you just need to know. Okay, so the nigrostriatal dopamine system, I've got cell bodies in the substantia nigra. They send axons way up front to the striatum, which includes the basal ganglia. Okay. Now here I've drawn just one neuron, one cell body in the substantia nigra. There's hundreds of thousands. Okay. All of these are going to kind of bundle their axons together. Okay, so there's a structure called the MFB. Median, sometimes you see medial, doesn't matter. Forebrain bundle. Okay, MFB is a large collection of axons going from the midbrain, these are in the midbrain, up to the forebrain. Now, I want you to focus on where you are. If I stick a needle into the substantia nigra and I excite this neuron, where is neurotransmitter released? So if I excite this neuron, is neurotransmitter released in the substantia nigra? Well, let's figure it out. Excitation of this will lead to an action potential. Where does the action potential go? It goes down the axon to the terminals. When I get to the terminals, that's where voltage-gated calcium channels open up, start that cascade of reactions, and I get the release of neurotransmitter. So if I stimulate the midbrain, the substantia nigra, the release of neurotransmitter is actually way up in my forebrain. This is another important concept. Where neurons get excited and where they release neurotransmitter can be a long way away. Are there voltage-gated calcium channels and synaptic vesicles in the soma, in the cell body? I haven't explicitly told you yet, but your guess, I hope, is no. It's only at the terminal where we have a synapse where we see those structures. Okay, so nigrostriatal. Clinically, this is very important because these neurons die in Parkinson's disease. Okay, so nigrostriatal dopamine, think movement and Parkinson's disease. The mesolimbic dopamine system is really important for reward. Okay, so when we think about addiction, this is the system we talk about. Here, the soma are also in the midbrain in a part of the brain called the VTA, ventral tegmental area. VTA. Okay, so the cell body's here. And we are going to send axons up to the limbic system. Now, you may not remember everywhere in the limbic system, but one thing I hope you remember about the limbic system is, yeah, there's a lot of brain regions. These regions are involved in motivation. They're involved in emotion. They affect pretty much everything we do. All right, so these neurons in the VTA are going to send their axons forward to the limbic system. Now, substantia nigra and the VTA are different parts of the midbrain, but they're near each other. So we don't reinvent highways. Okay, here we have a connection, this median forebrain bundle. So actually, the VTA neurons also are part of that MFB. Now, in terms of reward, I want you to think about the nucleus accumbens. This is key. This is also important for drugs of abuse. And we'll talk about that a little bit more. The last is a mesocortical dopamine system. And here we go from the midbrain up to the cortex. And when I say cortex, I mean cerebral cortex. Okay, so this mesocortical dopamine system is going to affect cognition. We are going to spend an awful lot of time talking about this. So anytime you see mesolimbic dopamine system, VTA, and nucleus accumbens, pay close attention and make sure you memorize that. That's going to be really important for you. Do me a favor, when you ask questions, please ask them to everyone, not just me. I'll answer this one, it's just fine. Isn't the nucleus accumbens in the basal ganglia? It forms part of the basal ganglia. Yes. So an area, the nucleus accumbens can actually be classified as being in two different divisions. Yes. Now, when I say the nucleus accumbens too, the nucleus accumbens is huge. So depending on which part of the nucleus accumbens we're talking about, there are different connections and different functions. So you're not confused. You're not confused at all. Okay. I'm gonna, instead of taking a break, I'm going to plow through and we are going to finish receptors. I'm going to go through this fairly quickly because I think you guys are probably getting tired and I'm getting tired of talking. So I'm going to make it through this fairly quickly and then we'll break. I'll be happy to take questions too. Okay, so receptors. There's two classes: ionotropic, metabotropic. Okay, ionotropic, as the name implies, probably has something to do with ions, right? Metabotropic has something to do with metabolism. Metabolism are the biochemical reactions in your body. Okay, so the names are going to help you out here. This is going to be a large protein that forms an ion channel. Ions flow through it. Okay, this is key. If I describe a receptor to you and I ask you, is this ionotropic or metabotropic? First thing, the only thing you need to ask yourself, do ions flow through the receptor? If the answer is yes, it's ionotropic. If the answer is no, then it's not. And the only other one is metabotropic, right? Okay, so ions flow through it. We're going to talk about two that we need to remember. One is a glutamate receptor. Okay. There's actually two. Don't worry about the subdivision now. There's something called the NMDA and the kainate. Okay, these are both glutamate receptors. Okay, and both of these are basically sodium ion channels. GABA is a chloride, is a chloride ion channel. We haven't talked about that at all yet. Okay, so the NMDA receptor is special. We're going to talk about that later. We'll focus on this kainate receptor, which is a sodium ion channel. Now, when we talk about the glutamate receptor and the GABA receptor, okay, what makes this a glutamate receptor is glutamate binds to it. Okay, so I'm going to draw this real quickly. So here is my receptor closed. Okay, now plus glutamate. Here's glutamate. Glutamate binds to it and changes the shape. Now it's open. This is a sodium channel. So now sodium can come into the cell. Okay, so when there's no glutamate bound to this receptor, it's closed. When glutamate binds to it, it opens up and sodium comes in. What do you think happens when this glutamate molecule leaves and is no longer bound to it? It goes back and closes. Okay, these have no refractory period or anything like that. If there's glutamate bound to it, it's open. If there's not, it's closed. Okay, so what's the effect on cell? My membrane potential, we're at rest, minus 70 millivolts. What happens? I've got glutamate here where this bar is. What happens? Sodium comes in and I'm going to get a depolarization. Now, if glutamate leaves and we close, eventually all those sodium ions that came in will diffuse away and will return to rest. Okay, so we get a depolarization. Now this moves me closer to threshold, right? That's what a depolarization is. Anything that moves me towards threshold is excitatory. Okay. Now here, I'm not going to draw it closed. Here we've got, let's say, GABA bound to my receptor. Now it's a chloride ion channel. Chloride is more highly concentrated outside than inside. So chloride's going to come in. What does that do? I'm adding negative ions in, right? That's going to hyperpolarize my cell. Again, over time, those are going to diffuse away and we'll go back. So I'm moving further from threshold. It makes it harder for me to generate an action potential. It's going to be inhibitory. Okay. Now, what makes both of these ionotropic receptors? Well, when neurotransmitter binds from them, they open up and ions pass through them, directly through them. Okay. Now, one thing real quickly, I want to remind you. This is my terminal. Right, here's my postsynaptic cell. If we're talking about a normal synapse, right, in the presynaptic cell, this is going to be the terminal bouton, the end of the axon. Where is this? This is going to be on the dendrite. Now, synapses can be other places. Okay, we have axosomatic, exodendritic, exoaxonal. Okay, but for right now, most of the synapses are going to be axodendritic. So an axon and a dendrite. Okay, so I'm going to move over here for a sec. So here's my cell. Poorly drawn, sorry. Here are dendrites. Okay, so I've got this axon coming in. So when we're talking about these voltage gated we're talking about these ligand gated channels opening, we're talking about a tiny little bit of the membrane. Okay, so here on the postsynaptic side, we'll get a small depolarization. Okay, so we'll have an effect here. Now, just because of physics and passive properties of the membrane, that's going to have a small effect on V sub M at the hillock. And remember, if we get greater than threshold, we get an action potential. Okay, so I'm bringing this up because you need to remember where you are in the neuron. When we're talking about voltage-gated sodium channels, voltage-gated potassium channels, action potential, we are only talking about the axon. When I'm talking about these ionotropic receptors, right, we're only talking at synapses. Now, this one little EPSP here, this one little potential, one little depolarization is going to have a very small effect on the axon hillock. But what you need to remember is there's 10,000 connections. So each individual one will have a very small effect, but when we add up all 10,000, we can have a huge effect. All right, I already used the word, but now I'm going to explain it. We talk about EPSPs and IPSPs. Okay, E equals excitatory, right? You see why it's excitatory, right? We're getting closer to threshold. This tells us where it is. So PS is postsynaptic. This P stands for potential, which is a separation of charge. Okay, so when we're talking about the glutamate receptor, this kinate receptor, it's a sodium ion channel. Sodium comes in, depolarizes the membrane. That gives me an EPSP, an excitatory postsynaptic potential. If we are talking about GABA, we have a chloride ion channel. That's going to make it inhibitory, an IPSP. So remember way back last week when we're looking at the action potential, right? We have this depolarization. And before I said it was magic. Okay, I said, don't worry about where that comes from. Now you should worry about where that comes from. Comes from this. Okay, so now you know where we get this initial little depolarization. It's from all these excitatory postsynaptic potentials, all these EPSPs. Okay, so ionotropic, first thing, does an ion flow directly through it? If the answer is yes, you're talking about an ionotropic receptor. Metabotropic, we're going to alter biochemistry. Okay, so here, if you look at this receptor, the way I've drawn it, there's no tunnel. Okay, so when neurotransmitter binds to it, it'll change the shape and it'll expose enzymes and other chemicals. Okay, so basically when the neurotransmitter binds, it activates enzymes. Which equals the start of biochemical reactions. Okay, now these are long chains of biochemical reactions. We're not going to get into too much detail about it. On these metabotropic receptors, on some of them, we classify them based on the names of those enzymes. Okay. We are going to talk about G protein coupled receptors. Okay, these are GPCRs. right? The reason I'm bringing this up and asking you to remember this name is when it comes to psychology, almost all of the receptors that are crucial for psychology are GPCRs. They're G protein coupled receptors. Okay, so the name of this first enzyme is just a G protein. It's just based on the structure of it. There's many different kinds of G proteins. I'm not going to make you memorize them all. All you need to know is this initial enzyme that starts this cascade of biochemical reactions is called a G protein. Okay, so any receptor that has this G protein is called a GPCR. And again, lots of drugs, both therapeutic and drugs of abuse, target GPCRs. That's why I'm talking about it. All right, so what ends up happening, and again, I'm really drawing this schematically and simplifying, okay? Our neurotransmitter binds and we get reaction 1. Okay, that's going to be exposure of that G protein, okay? So we'll get activity here. Then we'll have 2, and then 3, and then 4, okay? So we're going to get a series of products, a series of reactions. Now, this enzyme is associated with the receptor. The rest of these are free to float around the cell. Okay. Now, one of these could flow to a different part of the cell. Okay, so let's say 4 comes around and floats, and now it interacts with this protein. When it interacts with that protein, it might open it. And maybe that's an ion channel. Not a receptor, an ion channel. If this is a sodium, sodium could flow in. Okay? This product could go to the nucleus and affect DNA. We could go and change TH activity. Okay, there's literally thousands and thousands of different things these products could do. Okay, so while the neurotransmitter is bound to this receptor, we're making all these products. Those are going to go on and have their effect. When my neurotransmitter leaves, this biochemical reaction stops and we stop making these. Now, one thing you're wondering, if I make a whole bunch of this, even when my neurotransmitter leaves, won't this still be around? And the answer is yes. Okay, so metabotropic receptors have long-lasting effects. And by long-lasting, we're talking seconds to hours. When we talk about the ionotropic receptor, right? How quickly is that over? Think about the neuromuscular junction, right? As soon as that closes, we repolarize the cell really quickly. We're talking milliseconds, many milliseconds, but milliseconds. Okay, so metabotropic receptors are going to have long-lasting effects. That's good if you're studying biopsych, right? If you're studying biopsych, you want this stuff to last for a while. Things like mood. You don't want mood to be completely labile. You want it to be relatively steady. Okay, so that's a quick summary of metabotropic receptors. Hopefully when you refresh your memory on the chapter, it will make more sense. If you have any questions as you go, definitely email me, hit me up on the Discord server. I'm going to stop recording now and take questions.